This is an overview of an article entitled Single Nucleotide Polymorphism of the MLX Gene is Associated with Takayasu Arteritis, published in the October 2018 issue of Circulation, Genomic and Precision Medicine. Takayasu Arteritis, or TAK, is an autoimmune systemic arteritis with unclear etiology known to be associated with gender, race, and geographic location. The majority of TAK patients are young and middle-aged women of Asian background. The onset of the disease occurs between 10 and 40 years of age, and its prevalence is much higher in Asia than in Western countries. TAK lesions are mainly localized in the aorta and its major arterial branches. As arterial inflammation progresses with age, at the final stage of the disease, the entire blood vessel can be affected. A previous genome-wide association study, or GWAS, found that a single nucleotide polymorphism, or SNP, in the MAX-like protein X, or MLX, gene is significantly associated with TAK in Japanese patients. MLX belongs to the family of basic helix-loop-helix leucine zipper, or BHLH-ZIP, transcription factors, and is activated through formation of heterodimers with other BHLH-ZIP family members, including Mondo A. However, there is little information about the effects of MLX on the immune system. The risk allele of the GWAS SNP, RS665268, is a missense mutation causing the Q139R substitution in the DNA binding site of MLX. In the paper under discussion, the authors first assessed the effect of the SNP genotype on clinical characteristics of TAK patients. Among the observations was that the prevalence of moderate or severe aortic regurgitation, or AR, was significantly higher in patients with one or two risk alleles than in patients with no risk alleles. The mean number of arterial lesions was also significantly higher in patients with at least one risk allele. Based on structural predictions, the amino acid affected by the SNP, resulting in the Q139R mutation, is located in the DNA binding site of MLX, where it can interact with analogous DNA binding sites of other BHLH-ZIP transcription factors and potentially modulate heterodimerization. The authors hypothesized that the Q139R mutation, which increases the positive charge in the protein, enhances the formation of an MLX Mondo A DNA complex by creating complementarity to the negative charge in the same region in Mondo A. As shown in the panel on the lower right, the Q139R mutation does indeed promote the formation of the complex, as indicated by immunoprecipitation experiments. The authors went on to perform a series of experimental studies that found that the MLX Q139R mutation increases thioredoxin interacting protein, or TXNIP, expression through activation of the TXNIP promoter, promotes oxidative stress, upregulates NLRP3 inflammasomes, attenuates autophagy and enhances inflammasome formation in macrophages, and promotes the proliferation and adhesion of macrophages. Please refer to the online version of the paper for more details. In conclusion, the authors found that the MLK Q139R variant facilitates inflammasome formation, which in turn promotes the proliferation and adhesion of aorta-resident cells, a pathological manifestation seen in the aortas of TAK patients. Thus, MLX dysfunction may play a crucial role in the pathogenesis of TAK. These findings might have potential application in the development of novel therapeutic approaches for the treatment of TAK.